Thank you very much. Thank you for the introduction. Good afternoon, everyone. First of all, I would like to thank for this great opportunity that I can show you a quite interesting story that we are investigating at the Department of Microbiology at the University of Szeged. So the title is quite scaring, but I would like to start with a quite nice thing, and this is the fantastic fungi. The picture is showing here a picture from a movie made by Louis Schwarzenberg. And this is actually a movie about the fantastic creatures, the fungi, which are working as a digestive tract of the earth. So they are actually responsible for the recycling of the nutrition, and they are making the soil, which is extremely important to be able to live in the earth. And they are extremely diverse. We estimate that more than 5 million different fungi species exist. And actually the humankind took advantage already of this high diversity because we are using them as a food and we are using them for fermentation, for example, whiskey, beer, wine, and bread. Probably I used the wrong order, but anyway, they are also very important for the medicine because they're producing antibiotics. And why we are not aware about them that much? Because they are extremely hidden. We are not seeing them. They are living in the soil, inside the plants, and even inside us. Because it is well known that our microbiota has also fungal components called microbiome. And the different body sites, such as the gut, for example, has a different microbiota than the oral cavity. So as you can see here in this picture, the diversity is quite large and the composition is also different. And unfortunately for us, fungi can cause disease. And this disease-causing ability is sometimes linked to the collapse of the homeostasis of the microbiota. So even if you take a long-term antibiotic treatment, then bacterial diversity collapses and this opens the door for overgrowth of fungal species, and these ugly pictures can be taken because this is a Candida albicans infection caused in the mouth, and it's very uncomfortable, it's painful, and cause infection. Another very common disease in the oral cavity is cancer, and unfortunately the mouth cancer rates is increasing. And as you can see here in 2019, 53,000 new cases were identified in the United States, and Hungary unfortunately is the second in the rank, the total numbers of oral cancers in Europe. So a couple of years ago, we asked the question, could we link these two diseases together? And we performed a study in collaboration with the Faculty of Dentistry, and we analyzed the yeast carriage of healthy patients, healthy individuals, and compared that to oral cancer patients. OSCC means oral squamous cell carcinoma. As you can see here, the colony-forming units that we were able to recover from the oral cancer patients were extremely high. And if we went further and compared the healthy surface of the patient's mouth and compared that to the neoplastic surface, even in the mouth of the cancer patient, we were able to see differences. So I have to tell you one other information. If you treat oral cancer, then usually you use radiochemotherapy. And as this report shows that after the radiochemotherapy treatment, very often oral candidiasis took place. So this incidence is really high. It can be as much as 75%. So our question was, how cancer cells and candida cells are interacting? Is it possible that candida cause cancer? Let's say candida is carcinogenic. Or even if you have an established cancer, can candida worsen the outcome of the cancer, meaning increasing the invasiveness and helping the metastasis building capacity. We set it up a very, very easy experiment. As you can see here, we used cancer cells. And we caused a so-called injury. This is a wound healing assay. If you do a wound heal, if you do a wound, okay. Yes. If you do a wound and then look at the so-called healing, then you see that the cells are migrating with a straight line almost. On the next slide or video, you will see when we infected these cells with Candida albicans. Look at how the cells behavior changing. You will see a lot more single wandering cells, single migrating cells. They are deattaching and they are behaving completely differently. So we found this very interesting and went further, and we measured the matrix metalloproteases, which are responsible to loosen the cell-cell interactions or the cell-cell junctions, and we figured out that in the case of Candida infections, you have a much higher matrix metalloprotease activity in compared to the control. It is also well known that cancer cells are changing the metabolic activity, and there are so-called oncometabolites. We measured the metabolome of these infected cells, and you see two examples, the aspartic acid and the succinic acid, and both of them are increased in the presence of Candida infection. Then we analyzed the transcriptome. We isolated RNA, and then we made a total RNA-seq, and we analyzed how the gene expression change in the presence of Candida cells. This is a quite complex table, but what you have to appreciate is the red labeled genes, and these are the genes which were upregulated in the presence of Candida, and it's already published that they are linked to the invasion promoting effect in the case of oral cancer. Then we further analyzed the changes of the genes, and we analyzed the pathways, and we found many different pathways which are pro-tumor progression pathways. So not only on the metabolome level, but also the transcriptome level, we were identifying cancer invasion promoting events. But these all were in vitro. We wanted to see it in vivo, so we established a so-called Xenograft model. The Xenograft model means that we are introducing a human tumor in the mouse. And how we did that, we immunocompromised the animals, then we introduced a human tumor, and then we initiated an oral squamous cell carcinoma, and after 3 days, we analyzed the human tumor mass. So you can see here on this panel, the tumor was actually colonized with a lot of fungal cell. And then what we found was quite interesting. If you're analyzing this kind of samples, the pathologists can help you because they have so-called cancer markers, and these are 3 examples of cancer markers. The first one is the P63, which is a transcription factor. This is without Candida, and this is with Candida. If you see darker brown dots, that means the cancer is worse. Okay, and this one is the so-called E-caterin, which is a molecule which is important in the cell-cell interaction. Losing this molecule, losing the expression of this molecule means that the cancer is more invasive, and this was also the case in the oral Candida infection model. And vimentin is in opposite. So if you have more vimentin staining, then you have more aggressive tumor. And this was also caused by Candida infection. But someone can ask, okay, 3 days, that this is not a long time, it cannot be carcinogenic. So we then established another model, which is actually thought to investigate the real carcinogens like the smoking. So this 4NQO model is actually based on the carcinogenic effect of this substance. We put this in the drinking water of the animals for 8 weeks, and then for 12 weeks we initiated a Candida infection on these predisposed animals. And then we compared the carcinogen alone and the Candida albicans and carcinogen together groups. And this is a quite complex table again, but what I would like to highlight actually this one here. So if you look at this blue bar, this is actually the carcinogen alone. Let's say you are smoking. And this one here, the other one is the chance to get a cancer if you're smoking and you have if you have a Candida infection. So what we found in this experiment that having a Candida infection, if you have a predisposing effect, then you have 3 times more chance to develop a cancer without the Candida. So in summary, we showed that oral yeast infections can be a potential risk for cancer development, and oral cancer treatment actually increases the chance of Candida infections. And what is very important, that oral candidiasis can reduce the efficiency of oral cancer treatment because of the increase of invasiveness of the cancer. And Candida albicans infection can lead to poor prognosis in oral cancer. And I have to mention one thing. This is only the oral cavity. Our body contains Candida all over. For example, in the gut, it's very common there, and the vagina. So there are many other places or many other body sites where Candida can actually contribute the carcinogenesis. And if the cancer already established, then it can worsen the outcome. So we have to be aware about that. So I would like to thank for my group at the Department of Microbiology, the Fungal Pathogen Research Group, and I'm really proud of our National Scientist Academy students, Marton Horváth, who is actually already have a first author paper, and the second author paper is also accepted, and he just even started the MSc program, so I just can tell you that this program can be very successful. And our new student, Gergő Szvorény, and our already finished, but hopefully in the PhD program, reopening the Zsófia Tóth. And certainly we are really happy to have great collaborators from Hungary and from all around the world. And with this, thank you very much for your attention, and I'm really happy to answer your questions.
Thank you very much, and now I ask you to come forward with your questions. Don't be shy. That would be a very nice occasion to discuss with Professor Gácser what you still have in mind about this very burning issue. It's a very practical question as well.
So my question is that is there any other well-known fungi species which can increase the risk of oral cancer beside the members of the genus Candida?
Yeah, it's a perfect question. So actually, fungal infections in the oral cavity is mainly caused by Candida species. Not always caused by Candida albicans. There are other species which can cause oral candidiasis, but the majority of this disease is actually caused by Candida albicans. Any other fungi which can colonize our oral epithelia is much more rare. So if you would like to have a percentage that 99% of oral fungal infections caused by Candida species. Sometimes we see Saccharomyces infections, which is very surprising. But Saccharomyces can also infect the female genital tract. So very few cases were reported, but mainly candidiasis, which is predominant in the oral cavity.
Thank you for the answer.
Thank you. Any other question? While you're thinking about that, can I ask you that the therapy that is generally used against candidiasis, do they have any impact on tumor development or tumor generation?
Yeah, this is also a great question. So The main therapy is antifungals, azoles. This is the first choice, or in the case of Candida, nowadays they are using echinocandins, and they do not influence the cancer therapy at all. So that's why we also would like to suggest to use that. And one of the very interesting and promising collaboration with Norbert Pardy, which is just starting next week, we try to develop antifungal vaccine with them, which would actually then help to prevent this pro-cancer effect of fungal species. But this is just a start, and I hope next time when I got invited, I can talk about that more.
Thank you very much.